Good morning, children. In our previous class, we had studied about the light reaction of photosynthesis. Today, we will begin with the dark reaction. The second step of photosynthesis includes the series of reactions for which light is not necessary. So, you have to note that light is not necessary, and so it is called as the dark reaction. Its existence was indicated by R. Blackman, 1905, hence it is also known as Blackman's reaction. The reactions are biochemical in nature and are catalyzed by enzymes present in the stroma or matrix of the chloroplast. During this phase, that is the dark reaction, carbon dioxide that is obtained from the atmosphere is reduced with the help of reduced NADP, that is NADPH2, and ATP, which are produced during the light reaction. So what happens here, that in this phase CO2 that is obtained from the atmosphere is reduced, and with the help of reduced NADP and ATP, which are produced during light reaction, the end products of the reaction are starch or sugar. This fixation of carbon dioxide can occur either by Calvin cycle or C3 cycle or Hatch and Slack or C4 cycle. So now we begin with the Calvin cycle. Now what happens in the Calvin cycle is the main reactions in Calvin cycle, as the first product in a series of reactions is a 3-carbon compound, phosphoglyceric acid, the pathway is called C3 cycle. So you call Calvin cycle as C3 cycle because the first product that is formed in the series of reaction is a 3-carbon compound, phosphoglyceric acid. So, since it's a 3-carbon compound, this pathway is also called C3 cycle. The cycle is also said to be Calvin-Benson-Besham cycle after the names of its discoverers. The primary acceptor of carbon dioxide in this cycle is ribulose 1,5-biphosphate, RUBP. Calvin cycle can be divided into 3 distinct phases: carboxylation, glycolytic reversal, and regeneration of RUBP, and it takes 6 turns of Calvin cycle to generate 1 molecule of hexose or sugar. So now we begin with the first step in the Calvin cycle, and that is carboxylation. Now it is the fixation of carbon dioxide into a stable organic intermediate. The first step in Calvin cycle is carboxylation of ribulose 1,5-biphosphate RUBP by atmospheric carbon dioxide in the presence of enzyme RUBP carboxylase or Rubisco. 1 molecule of RUBP joins with 1 molecule of carbon dioxide to form a 6-carbon unstable compound, which is quickly converted into 2 molecules of 3-phosphoglyceric acid or 3-phosphoglycerate, PGA, a 3-carbon compound, with the help of an enzyme RUBP carboxylase, also called carboxydimutase. The next step is glycolytic reversal or reduction. These are the series of reactions that lead to the formation of glucose. So here you can see the formation of glucose. The steps involve utilization of 2 molecules of ATP for phosphorylation and 2 molecules of NADPH for reduction per CO2 molecule fixed. For conversion of 3-PGA or 3-phosphoglycerate into triose phosphate, ATP and NADPH are required which are supplied by the light reaction of photosynthesis. These steps in fact are essentially a reversal of glycolysis. However, reducing power is obtained from NADPH rather than NADH. For removal of one molecule of glucose from the pathway, fixation of 6 molecules of CO2 and 6 turns of the cycle are required. Now we come to regeneration of RUBP. Regeneration of CO2 acceptor molecule RUBP is crucial if the cycle is to continue uninterrupted. The regeneration steps require 1 ATP for phosphorylation to form RUBP or ribulose 1,5-biphosphate. This phase involves many steps and enzymes. For the continuous function of Calvin cycle, there must be a regular supply of ATP and NADPH. The fixation and reduction of one molecule of carbon dioxide thus requires 3 molecules of ATP and 2 molecules of reduced NADP coming from photochemical reactions. For the fixation of 6 carbon dioxide molecules, the reactions of Calvin cycle can be summed up in the following overall equation. Now see the equation: 6 RUBP plus 6 CO2 plus 18 molecules of ATP plus 12 reduced NADP plus 12 molecules of hydrogen ions give RUBP plus 18 ADP adenosine diphosphate plus 18 inorganic phosphate PI plus 12 NADP plus glucose C6H12O6 plus water. The compound 3-phosphoglyceraldehyde holds a pivotal position in the cycle. It may be transported out of the chloroplast and converted into various sugars and cell wall carbohydrates, or may move into the metabolic pool. So, this is the Calvin cycle.